Okay. Well, so welcome everyone back for the third and final lecture in this series that Alison will be giving. Um, and just before, I'll send in the chat a link to the slides and to the previous lecture material. And during the course of the talk, if you have questions, you can unmute yourself and ask, or put it in the chat if it's not something that's immediate and necessary. Okay, so we will continue with Alison's third talk on some mathematical models of evolution, the infinitesimal model.
Great, thank you, Ivan. So third lecture, third model, but still actually the underlying motivation or the application I'll try to get to anyway is to understand something about the interaction between natural selection and spatial structure. But this time we're going to be thinking of natural selection as acting on a trait rather than acting on just the allele at a particular locus. And this will require a new class of models for us. And to understand the origin of those models, one has to go back a very long way. But first, let me do my thanks. So you've seen these two before, Nick Barton from IST Austria and Amandine Weber, now at Paris 5, two of my favorite collaborators. And we've been moving backwards in time. So the model that I'm going to talk about today actually began with the modern evolutionary synthesis, which is over 100 years ago. And we're going to begin by talking about that modern evolutionary synthesis. So the modern evolutionary synthesis is really the origin of theoretical population genetics. It's when the subject began. And it's when the work of, on the one hand, Darwin, and the other hand, Mendel, was finally reconciled into a single theory. Now, I know I don't really have to tell you what Darwin's theory of natural selection says, but let me anyway. So Darwin tells us that heritable traits that increase reproductive success will become more common in a population. And for that to work, it requires that there's variation in the population. If everyone's the same, no one has an advantage over anybody else. And offspring must be similar to their parents. And both of those features are provided by Mendel, because Mendel tells us that traits are determined, well, at least strongly influenced by genes. They would have said gene. The word gene is comes much later. At the time of the modern evolutionary synthesis, they called them Mendelian factors. And these genes occur in different types. We called them alleles in previous lectures, and that gives us the variability that Darwin needs. And offspring inherit genes from their parents, so that gives us the heritability. But in fact, it took a very long time for the two theories to come together, roughly 50 years after their publication. And one of the reasons for that might perhaps be that Mendel concentrated on the big differences that arose from changes at single genetic loci, like the difference between a yellow pea and a green pea, or a wrinkled pea and a smooth pea. Whereas Darwin much more focused on how natural selection acting on very small changes could accumulate over time to produce a very big change. But the two theories were united, and in particular, they had to be united by showing how we could understand complex traits like height and weight and so on in terms of Mendelian factors. And it was really Fisher who in 1918 showed that the observations that had been made by biometricians who like to measure families, so they'd measure the heights of parents and then the heights of their offspring and things like this. So Fisher showed that these observations were actually consistent with these complex traits being the result of a large number of Mendelian factors, each of small effect, plus some environmental influence, obviously. So the very simple model you might have at the back of your mind, which we will return to later, is to think of M genes, so lots and lots of genes, M's a large number, each with two alleles of effects plus or minus 1 over root M, say. And then the genetic component of the trait value would just be some underlying number Z0 plus a sum of eta L over square root of M, where eta L is plus or minus 1 with equal probability. And of course, that's just normally distributed. And so the normal distribution that we see in many of these complex traits arises naturally if you adopt Fisher's model. And Fisher's infinitesimal model showed us how to propagate those trait values, those complex trait values, or at least their genetic component through time. So under Fisher's infinitesimal model, the trait value of the offspring of two unrelated parents is going to be the mean of the parental trait values plus a normally distributed error. And that normally distributed error is going to have mean 0 and variance given by what is called the additive genetic variance. And we'll see what that is as we progress through the lecture. Okay. And then Fisher's fundamental theorem of natural selection says, well, the rate of increase in mean fitness in a population is proportional to that additive genetic variance in fitness. So it's the additive genetic variance on which natural selection is able to act, and that's going to be important. Okay, and this is a quote I very much like, and we'll come back to it later, again due to Fisher, who said that natural selection is a mechanism for generating an exceedingly high degree of improbability. So perhaps the way in which those Galapagos finches in the bottom right move from having a very narrow beak to a very broad beak through these different stages. Okay, so the basic model then is that the trait value is going to have a genetic component and a non-genetic component. And for today's purposes, we're going to ignore the non-genetic component. It's actually very important to make the maths work smoothly. So some of what I say is not strictly speaking true mathematically, but it becomes much more true if we add on an environmental component, perhaps in the form of another normally distributed random variable. And the genetic component is normally distributed, and the mean is the average of the values of the parents. And in a large outcrossing population, so outcrossing just means that you always mate with somebody who is completely unrelated to yourself, then that V0, the variance of the genetic component, is constant. Now, if it's not an outcrossing population, if I might be related in some way to my mate, then that variance is going to decrease, and it decreases in proportion to relatedness in a way that we're going to make a bit more precise. Okay, so in that simplest case, though, where the variance is held constant, if the mating is purely random, which corresponds to a trait being neutral, and you just think of individuals as choosing their mate just uniformly at random from everyone else available in the population, then what Bulmer showed in 1971 was that the trait distribution in the whole population under this model will rapidly converge to a Gaussian. And that Gaussian will have variance twice V0. So V0 was the genetic variance within family, and the variance of across the whole population is just going to be twice that. And it's informative, I think, to think about why that might be true. So let's use V1 to be the variance in the parental population. Well, then the variance among offspring has two components. The first is the variance that arises from the way in which the parents were chosen, so from the Z1 plus Z2 over 2. And the second is the variance V0, that is the additive genetic variance that we've supposed is reproduced in every generation. Now, the variance of Z1 plus Z2 over 2, well, that's just going to be half the variance of one of the Z's, right? Because we get a half squared is a quarter, but then we've got Z1 plus Z2, which are independent picks from the population because we're looking at neutrality. So that gives us a quarter times 2 times V1, and that gives us V1 over 2. So at equilibrium, we're going to see that V1 is twice V0. And so half the variance that we're seeing in the population is coming within families and half between families. Okay. Now, I should emphasize, and I can't emphasize this enough, that in general, the infinitesimal model, as I'm going to define it, only says that the genetic components within families are normally distributed. It does not say that the distribution of a trait across the whole population is going to be normally distributed. And indeed, it could be very far from normal. It could, for example, be that my population is subdivided in some way. But the infinitesimal model is ill-defined, and so I'm free to define it as I wish. So the way I'm going to define it is that trait distributions within families are normally distributed, and the variance-covariance matrix that across those families is going to be determined entirely by that in some ancestral population, and by things that we will call the probabilities of identity. And the probability of identity of two individuals just says, if I sampled a gene from me and the corresponding gene from Ivan, what's the probability that those genes are inherited from a common ancestral gene, however many generations ago my ancestral population lived? So that would be the probability of identity of our genes. And those probabilities of identity are determined by what we're going to call the pedigree, and we'll talk a little bit more about the pedigree as we go forward. But what's nice about this model is that as a result of the multivariate normality, if I condition on knowing just some of the trait values within a pedigree, then that has predictable effects on the mean and variance within and between families. And this is a property that's been exploited by animal breeders for many, many centuries. Okay, so let's talk about pedigrees a little bit, because in the previous lectures, we've concentrated on haploid populations in which each individual is carrying one copy of each gene, and each individual gene we think of as having a single parent in the previous generation. And so we were able to characterize the ancestry of our populations in terms of genealogical trees. But now I want to think about populations in which individuals have two parents. But because I'm intrinsically lazy, I want to consider populations that I don't have to subdivide into males and females. So we're going to think about hermaphrodites. So there's a nice picture of a snail to remind you of that there. And in the simplest possible model of inheritance in this diploid world, in this world in which everybody carries two copies of each gene, the population size is held constant, exactly as it was in our original haploid right Fisher model that we've talked about before. But now every individual, and I'm representing individuals by these large circles here. So a large circle represents an individual, and every individual has two parents, which they choose independently at random from the previous generation. And if I haven't made any mistakes, there is a blue line, or there are two blue lines connecting each large circle to two large circles in the previous generation. Okay, and in this picture, time runs down the picture, so going into the past is going up the picture. And I claim that these three individuals in this particular cartoon are ancestral to everybody in the current population. Now, it's not terribly easy to see that, so I did some coloring in for you. So what I've done is I've colored in that individual in the top right, cyan, and I've just traced the descendants down to the current population. And you see that everybody in the current population you can trace a route back through this ancestry to get to the cyan individual in the ancestral population. Now, of course, if I'm only carrying two copies, if I look at a single individual down here, and they're carrying two copies of a particular gene, but I'm telling you that there are three individuals in my pedigree that are ancestral to everyone down here, so in particular ancestral to this individual, they can't all be contributing genetic material to that individual. And just to check that, so pedigree ancestry must be different from genetic ancestry. And what I didn't tell you before is that the small circles are meant to represent an individual copy of each of the genes. So there are two small circles within each large circle. And when I trace, so one will be inherited from each parent, and the choice of the small gene within the parent is just, again, uniformly at random. Okay? And what you can see is that this cyan individual didn't actually contribute any genetic material to the current population, because what I've done is I've used red to trace genetic ancestry. So although this individual is in the pedigree ancestry, at this particular locus, it doesn't contribute any genetic material to the current population. Okay. Now, I can recover the genealogical trees that we've talked about for the Wright Fisher model just by tracking the genetic material. So I can recover my haploid Wright Fisher model by just tracking the ancestry of individual genes, and in fact separating them into diploids is immaterial. But that's as we see, is not something we're going to be able to do in general when we're interested in following selection acting on traits. But what we see in this picture is that if I track the genetic material just corresponding, because it was easier to draw, to the leftmost of the genes in each of these individuals in the current population, they've not reached a common ancestor by this time here, when these three guys were ancestral to everybody in the pedigree. So pedigree ancestry is happening faster than genetic ancestry. And there's a nice result on that due to Chang. So this dates back to 1999. And what Chang says is that the most recent pedigree ancestor of a population of size n is, with the probability tending to 1 as n tends to infinity, just log 2n generations ago. And it's an even cuter result, which says that if I trace back 1.77 log 2n generations, then everybody in the ancestral population is either a pedigree ancestor of everybody in the current population or of nobody in the current population. So it's a remarkably tight result. I mean, this is really homing in on the pedigree ancestry, but it's happening over a very, very different timescale to the timescale that we saw for Kingman's coalescent, which was the timescale of genetic ancestry, which was happening over n generations. So pedigree ancestry and genetic ancestry are very different. And one of the reasons for that is that there are many, many different routes through the pedigree. Okay. So how am I going to think about these pedigrees? The picture so far, it's just been a mess, right? You've got all these crossing lines everywhere, and they don't look particularly comfortable to work with. And it turns out that a nice way to think about pedigrees is to think about matrices, to represent them as products of matrices. And I have to issue an apology here. Time in the next couple of pictures is running in the opposite direction. This is a very ecologically friendly talk, which has recycled some old pictures. And unfortunately, my new computer doesn't have the software to edit the old pictures. So I was unable to turn everything upside down. So here is the present in these pictures. It's still 5 individuals. Here's the present. And there is now just, I've only tracked the whole individual, not the individual genes within them. And each individual is connected to 2 parents in the previous generation. So here's the present. We trace back into the past. And as I go backwards, I'm also going to allow self-fertilization. It's just convenient to do so. Okay. So how am I going to represent that pedigree as a product of matrices? Well, the pedigree spanning T generations, I claim, is a product of, is just the random matrices, just T different random matrices. And the way I define them is that the matrix MT is going to tell me about the parents of individuals in generation T. So the ith row of MT is going to tell me about the parents of the individual that is labeled I in generation T. And those parents, of course, lived in generation T+1 before the present. And here I've just taken population size to be constant. There's no need for the population size to be constant. It can vary as I wish. But because we've been talking about the Wright-Fisher model, let's stick with the constant population size. So here's the present. So this is meant to be the matrix M0. And for example, the individual labeled 1, their parents in the previous generation were labeled 1 and 3. And so in my pedigree matrix, the first row has got a half in the first position and the third position, and otherwise zeros. And similarly, say the last individual, the individual labeled 5, their parents were the individuals labeled 4 and 5. And so in the bottom row of the matrix, there's a half in the 4th position and a half in the 5th position. Okay. And the half is just Mendelian inheritance. And when the selfing, I just substitute a 1. So for example, here, the first individual, this was the 3rd generation in my previous picture, and the first individual selfed. So both parents were labeled 1, and I just put a 1 instead of having 2 halves at that point. Okay. Now, haploids or diploids. I hinted that this was going to matter. For the sort of models we've talked about so far, where we're just talking about a single genetic locus, it doesn't usually matter whether we think about a haploid model or a diploid model, because we can either track from haploid phase to haploid phase in reproduction, or from diploid phase to diploid phase. So haploids mate to produce diploids. So they fuse to produce diploids. There's immediate meiosis, and then we go back to haploids. Or you start with a diploid, and that splits into haploids during meiosis, but after reproduction, they immediately fuse back into diploids. So I can either follow the gamete phase, or I can follow the diploid phase, and it doesn't really matter which I do. And one can usually interpret selection acting on diploids in terms of what it's doing to those gametes. So I can interpret it as some action of selection on haploids. But whether I look at haploids or diploids really does matter if I'm looking at selection on a trait, because selection on a trait is conditioning on the trait is providing information about the whole genotype. So I need to know what's happening in both chromosomes, if you like, in the adult individual. Okay, now I said that in our version of the infinitesimal model, we're going to have the variation within families, so within offspring of 2 individuals sampled from my population, is going to be normally distributed. So this is just to say, if I sample an individual from among their offspring, its trait distribution is normally distributed. But the variance of that distribution was going to decrease in proportion to the probability of identity. So I ought to tell you about the probability of identity. And the probability of identity, as I said before, is just the probability if I take 2 homologous genes, so 2 genes at the same point, so 2 corresponding genes, in individuals labeled I and J, and I ask if they're alive in generation T, and I ask what is the probability that those 2 genes are descended from a common gene in the ancestral population, that probability is what we call the probability of identity, and we denote it by F for historic reasons. So what one can do is write down a recursion for the probability of identity. And let's just do that in the haploid case. I've written the diploid case underneath. It's not much more complicated, but let's think about the haploid case. So if I'm haploid, I carry just one copy of this gene, but I was produced by diploid reproduction. So I had two parents. Similarly, the individual labeled J had two parents, and with equal probability, they inherited the gene in question from the first of those parents or the second of those parents. So there are a total of 4 parent pairs that matter to me, because those are the possible ways in which the gene can have been passed down from the parents to this current day individual. The way that the gene in individual I and the gene in individual J can have arisen, there are 4 different ways of that happening. And how can I identify those? Well, in my pedigree matrices, remember that MIK would have a half in the kth. This would be a half. So my ith row would have a half in the kth position if the individual labeled K in the parental population was a pedigree ancestor. And similarly over here, the jth row of my pedigree matrix would have a half in the elfth position if the elfth individual in my parental population was a pedigree ancestor. So when I multiply these two things together, I'm going to get a quarter for each of the possible pairs of parents. Okay. And Mendelian inheritance says the probability that I inherit my genes in individual I and individual J from any one of those pairs is precisely a quarter. And for each of those patterns of inheritance, I then say, aha, well, if I know that I inherited this gene from this parent, from the parent labeled K, and this gene from this parent labeled L, then the probability that my genes are identical, so inherited from a common ancestor in the ancestral population, a common gene in the ancestral population, is just FKL of T minus 1. And so now I have a recursion which tells me how to write down the probability of identity of the two genes in individual I and individual J in generation T in terms of the identities in generation T minus 1. So that's just a very, very convenient way to represent pedigrees. And these matrices in large populations are very sparse. So actually, numerically, they're quite easy to handle, or at least they're quite easy for my collaborators to handle. Okay, and there's a slight twist in the diploid case because you have to worry about selfing. Okay, so what does the infinitesimal model as we understand it know? So until recently, the infinitesimal model wasn't really written down anywhere. Nobody wrote down what they meant by the infinitesimal model. We were all meant to know. And Nick and Amandine and I decided that we would write down once and for all what we considered the infinitesimal model to be, and then we would try to justify it as a model. So what we're going to do is we're going to write P of T for the pedigree relationships between all individuals up to and including generation T. So if you like, that's just those matrices. And we're going to write Z of T for the traits of all individuals in a pedigree up to and including the Tth generation. And the quantity that we've got a statement to make about is the difference between the trait values of the individuals in generation T. So that's this vector Zj is going to be the trait value of the individual labeled J in generation T. And there are NT such individuals. It can vary from generation to generation. But if I center that trait value by subtracting the mean value of the traits in the parents of that individual, so the square brackets will always denote parents, and it's convenient to think of them as being parent 1 and parent 2, which is indeed how my son refers to us. So maybe it's the right way to go. So Zj1 is the trait value in parent 1, and Zj2 is the trait value in parent 2, and this is the mean. So centering on that mean, our claim is that even if we condition on knowing the pedigree all the way up to the current day, and we condition on knowing all of the trait values in the pedigree up to and including the parental generation, then this vector is approximately not a multivariate normal. So we're not saying that if you look at the trait Zj, we've got a normal over the whole population, they're normally distributed. We're saying that if we look within families, these traits satisfy a multivariate normal, and there is a diagonal covariance matrix. And that covariance matrix, those entries, the jth entry in that diagonal is going to be the segregation variance among offspring of the parents of individual J. And what do we mean by that? Well, we mean the variance in this ancestral population multiplied by 1 minus the probability of identity of the genes that we're taking out of our parents. Okay, so we'll see why this makes sense in the next couple of slides. Okay, so why might this be a reasonable model? Let's think
Can
about.
I ask you a question?
You may.
So the idea, I guess, is that you have this pedigree, and given pedigree, you have some. Well, on top of that, there's the randomness of how of who, who inherits which genes from which
Exactly
ancestors. So, but wouldn't that then induce a correlation between the behavior, between these ZJs, I guess, or maybe I'm not understanding.
Be patient, Professor Corwin. Be patient. Okay. It all will be revealed. I'm coming to that, I promise. Let's see why you shouldn't worry, or you should worry, but you shouldn't worry too much. I did tell you nothing I wrote down would be strictly true today, but it's almost true. So why is it almost true? Well, let's think about additive traits and haploids. So we're not going to think about mutation. We're just going to think about the genetic component of a trait, where the genetic component is determined by a large number of unlinked loci. That just means the Mendelian inheritance at each locus is going to be independent. I don't want to think about loci that are close together on the chromosome. And we're supposing that at each of those loci, the effect on the trait value is small. So we're going to scale it by a 1 over square root of m. And these are going to be taken to be mean 0 and order 1. So eta JL is going to be a scaled allelic effect at locus L in the jth individual. So I'm always going to try to use J's and K's for individuals and L's and M's for loci, genetic loci. Okay. And Z0 bar is the average in the ancestral population, and that's why I can take these etas to be mean 0. Okay, so that's the trait value in individual J. And in the ancestral population, I'm going to suppose that for different individuals, the scaled allelic effect at locus L is independent. And it's just sampled from, we just sample independently at random from some distribution of trait values in the, scaled allelic effects in the ancestral population. And it can be different for different loci, evidently. Okay, so the key thing is these are independent for different J. All right, so let's see what happens during reproduction for this model. So remember that 1 and 2 refer to the first and second parents of an individual. So eta JL1 is going to be the scaled allelic effect at locus L in the first parent of the jth individual. And similarly, ZJ1 is going to be the trait value of the first parent of individual J. And now we're going to represent that Mendelian inheritance that Ivan just mentioned by using Bernoulli random variables. So I'm going to write XJL to be 1 if the allelic type at locus L in the jth individual is inherited from the first parent of that individual, and otherwise it's 0. A Mendelian inheritance tells us that the probability it's 1 is a half. The probability I inherit that type from my first parent is a half, from the second parent is a half. This is a haploid model, so it's very simple. Okay, and then I can write the trait in the offspring. Well, I've got the Z0 bar still, plus, now what do the scaled allelic effects look like? Well, if I inherited my allele at locus L from the first parent, then XJL is equal to 1, and there's no mutation, so I'm inheriting exactly that allele, and so my type, eta JL at that locus, is going to be eta JL1. So this is what happens if at that locus I'm inheriting from the first parent, and that happens at probability a half. And if I inherit from the second parent, then XJL is 0, so I don't see this term. 1 minus XJL is therefore 1, and so I see the allelic type inherited from the second parent. So all this is doing is tracking the Mendelian inheritance. This Bernoulli random variable is tracing the Mendelian inheritance, and I can rearrange this expression, and I want to do that to justify the mean of my random variable being the mean trait value. So I can take out ZJ1 plus ZJ2. So ZJ1 over 2, it wasn't up here, so I better subtract it off somewhere in this expression, and that's that half times minus eta JL of 1. ZJ2 over 2, well, actually, ZJ2 occurred as a whole in a whole copy in this expression, because I had a 1 here multiplying e to jl of 2. So I better add on another half, and that's that half times this. And then the xjl terms are just copied from the line above. Okay. And notice in particular, this was what we wanted as our mean. And this is indeed a mean zero random variable in the sum, because Mendelian inheritance is independent of the allelic types in the parents. And my Bernoulli random variable has got mean a half. So this sum has certainly got mean 0. Okay. And I am claiming that this thing is approximately normally distributed, and that the variance is going to be given by this quantity related to the variance in the ancestral population. So suppose that I could convince you that the distribution of e to jl of 1 was not much affected by knowing the trait value in parent 1, or indeed any of the ancestors of that trait. Well, if I could convince you of that, then e to jl of 1 is just a sample from the ancestral population. Now, if my if e to jl of 1 and e to jl of 2, if I'm seeing identity, if they're inherited from a common parent, from a common ancestor in the ancestral population, then the contribution here is going to be 0. If they are inherited from distinct parents in the ancestral population, because we declared that distinct individuals in the ancestral population were sampling independently from this background allele frequency, the variance of this term is going to be twice the variance in the ancestral population. Okay. Now remember, this guy is independent. This is Mendelian inheritance. It doesn't know anything about trait values. And this was mean 0, and its variance is just going to be a quarter. So altogether, this expectation squared, and I've got this slightly wrong, of course, is going to be, so the expectation of this whole thing squared is going to be a quarter times whatever the expectation of this guy squared is. And the expectation of this guy squared, we just said, was going to be twice this variance if I had independent picks from the ancestral population and 0 otherwise. And that was precisely what this whether or not I had independent picks in the ancestral population was determined by this probability of identity. And so putting all that together, I'm going to see that the variance of this sum is a quarter from my Mendelian term times 2, gives me a half, times the variance in the ancestral population. Well, that's twice V0, twice the within family variance, times 1 minus the probability of identity. So if I can show this bit, if I can convince you that e to jl1 is really just a pick from my ancestral allelic types, then I hope I've convinced you that at least approximately, given that I've got a sum of 1 over root m, sum 1 to m, you can see a central limit coming theorem coming in, that approximately I've got the right distribution if I take a normally distributed random variable with these, with mean given by this quantity and variance given by V0 times 1 minus the probability of identity. So what I need to do is convince you that knowing the trait value doesn't tell me much about what's going on as a particular locus. So what we're going to do is going to go back to our toy example and see if we can believe it in that setting. So let's suppose that the ETRLs are IID with ETRL plus or minus 1. So those are my scaled allelic effects. And then the trait value in an individual, I've dropped the subscript j because I just want to look at a single individual, is just given by this expression where ETRL is plus or minus 1 with equal probability. And I've taken Z0 bar to be 0 because it's kind of, you know, it's just a normalization. It's not actually intrinsic to the maths. Okay, well, in that very simple toy model, if you tell me that the trait value in the parent, it can only take the form k over root m for an integer k, and if you tell me that the trait value in an individual is k over root m, if you tell me the trait value is k over root m for some integer k, then I can write down explicitly the probability that the allelic type at the first locus, for example, is equal to 1. Now, I've chosen m and k to be even to make the calculation beautiful. And then what happens is, well, it's just Bayes' rule, right? So the probability that this sum is equal to k, given that e to 1 is equal to 1, is the same as the probability that the sum, so I rearrange this, right? I write it in the other order. So I want to know the sum is equal to k given e to 1 is 1, divided by the sum is equal to k, multiplied by the probability e to 1 is 1. And the sum that this is k given e to 1 is 1, well, that's just the sum that the remaining m minus 1 of them add up to k minus 1, right? Now, for each of the possible ways of writing down a string of plus and minus 1s of length m minus 1, such that that string adds up to k minus 1, because eta L was plus or minus 1 with equal probability, that string will have probability 1 over 2 to the m minus 1 for any such string. And similarly, the probability of any string of length m, in which the sum of the minus 1s and 1s is k, will be 1 over 2 to the m. So that's these terms. And then I've just got a combinatorial factor. And the combinatorial factors are these binomial coefficients. And out front, remember, I've got this probability e to 1 equals 1. And if you multiply out all the factorials, you discover that this ratio actually simplifies and is 1 plus k over m. Okay. So what we've shown is that if the scaled allelic effects are just these IID Bernoullis, then the distortion of the probability that e to 1 is 1 is 1 plus k over m. Well, remember the trait value was k over root m. So for a typical trait value, k over m is going to be on the order of 1 over root m. So actually, I'm getting very, very little information about what's happening at locus 1 from knowing the trait value in the parent. Now, if you tell me the trait value is extreme, if you tell me the trait value is exactly m, then of course I automatically know that e to 1 is 1. But as long as you don't go too extreme on me, I don't get very much information. And the reason that for typical k, the distribution of e to 1 is almost unchanged is, of course, because there are so many different configurations of allelic effects that correspond to the same trait value. And one can extrapolate from that and have many, many different combinations of allelic effects, which will give me the trait values that I declare on my pedigree. And as a result, it is not ridiculously difficult to prove that this quantity is indeed approximately a mean zero multivariate normal, at least as the number of loci tends to infinity. And the error that one makes, as long as one doesn't choose too extreme trait values, in the sense that you can see how to make precise from the previous slide, you need that the trait value divided by root m is still small. And provided the segregation variance doesn't get too small, which corresponds to saying, provided I don't have too much inbreeding in my pedigree, then this approximation will be valid for the order of the square root of m generations. And for an animal breeder, that's absolutely fine. That's a very long time. Okay, so that's our infinitesimal model. And what I want to do is try to use this infinitesimal model to say something interesting. And if you thought the mathematics was hairy so far, it's about to get a whole lot worse. But let me tell you about the situation that I want to just make some very crude calculations about. I'm not pretending that this is a project which is in any sense finished, although there's a published paper on it. It's a project which suggests that one could do some really interesting things with the infinitesimal model and extensions of it. So here's the situation that I'm modeling. I am modeling a population which is, there's a large source population living in this cold, wet, mountainous region, and it is trying to colonize a desert island. It's trying to colonize a new habitat to which it is not well adapted. So here we have the population of Northern Europe trying to colonize the Spanish-owned islands off the African coast. And in order for them to, the migration is needless to say one directional. And in order for them to really thrive in this new environment, they have to adapt. They have to learn to live in a warm climate and learn to speak Spanish. So the questions that one might ask about this situation, this isn't such a ridiculous biological situation, it happens all the time where you've got a source population that's trying to colonize new territory. And the sort of questions one might ask are, well, what's the chance that a single migrant can actually establish a population? What's the time to establishment if I have a steady number of migrants arriving? How long before I actually get a well-adapted population, a population that really is adapted to living under these new conditions? And if there's a stationary distribution of trait values on my desert island and a stationary distribution for the population size, what does it look like? Okay. Now, I'm not quite going to answer any of these questions, but I'm going to give you some indications as to interesting things that do happen in this situation. But first, let's modify our infinitesimal model to make it work in this setting. So we've got a large source population, and we're going to suppose that the trait values are normally distributed with mean ZS bar, so S for source, and variance twice V. So remember, Bulmer told us that everything was neutral. It would all look pretty much normally distributed. Now, I apologize that I've used M for migrants when before I used M for numbers of loci. That was me not paying attention. So now I'm going to suppose that M migrants enter my island population in each generation, and those migrants are unrelated to one another. Okay. Now, if I write N of T for the population size in generation T on my island, and Z bar of T for the mean trait value, I want to know how N of T and Z bar of T evolve with time. So before the new generation of migrants arrive, I'm going to suppose that the next generation, so the number of offspring produced by the population already living on the island, is Poisson, and it's going to have expectation N of T times W bar. What's W bar? Well, W bar is going to be the mean fitness of offspring that are produced across random matings in this island population, and fitness is going to be measured just in terms of breeding value. Okay, so how many offspring am I going to produce? And so if W bar is less than 1, then this population is really only maintained by immigration. So we're really interested in populations which manage to achieve a mean fitness which is bigger than 1, so that they take off and they're really established, and we've got a stable population that can go off and grow on its own. It doesn't depend on migrants to survive. Okay. So I'm calling this Nick's model because the mathematics becomes so hairy that I really don't want to accept any responsibility for it. So we're going to use the infinitesimal model. So offspring of individuals I and J have a mean trait value given by the mid-parent value and a variance. And the variance, it depends whether we've got haploid or diploid. So I showed you how to derive this kind of expression for haploid inheritance. Please believe me, you can do the same for diploids, and the segregation variance will be given by a subtly different expression. It'll be given by this expression, but I didn't want to go through it, but it's really no more difficult. And so if I assume that the fitness of an individual with trait value Z is e to the beta Z, then how big is N of T times W bar? So how many offspring am I producing? How are the island dwellers producing to feed the next generation? Well, there are, remember, we're taking the average of W bar as an average over all possible random matings between individuals. And because we allow selfing, there are N of T squared possible random matings. So I would have a 1 over N of T squared here, but I'm multiplying by N of T, so that gives me 1 over N of T. And then this sum, if I weren't so lazy, would be sum ij equals 1 to N of T. And then for each random mating, remember, we're saying that the offspring of individuals I and J have a mean trait value given by the mid-parent values. That's this Zi plus Cj over 2, and a variance given by the segregation variance. And if I've got a normally distributed random variable with mean Zi plus Cj over 2 and segregation variance Vij, then the expectation of e to the beta Z is given by this expression. So that's where this comes from. Okay. Now, what we would expect, of course, is that density-dependent fitness and stabilizing selection would ultimately limit the population size, but presumably, by the time that happens, the population is established. So before stabilizing selection and some sort of perhaps logistic control of the total population size really kicks in and changes things, we're assuming the population is established. So we're going to stick with this very simple model, which is equivalent to taking a branching process model, if you like, because it's easier to work with. Okay. It'll be hard enough. So the first question we asked when I drew a cartoon of the situation with the source population and the island population was, well, could a single migrant establish? So instead of having steady state migration, if I just have a single diploid migrant with trait value Z0, can it establish a population? And because I'm allowing selfing, it can at least produce offspring. And on the left-hand side, we've got a graph which is on the vertical axis. This is the probability that I reach a population size of at least 100, which I'm going to declare to mean I've established. And on the bottom, this should be a ZS bar, but because Nick produced the picture, I can't edit it, or I could, but it would take me a long time. So imagine there's a bar over here. So this is the mean trait value in the source population. And we're supposing that's negative, and that, you know, in order to get growth, you need a trait value of at least 0. So we're really asking, you know, what's the chance we can overcome the fact that we're really poorly adapted to our environment, but we've got adaptation going on, and we've also got a certain randomness because our immigrant was sampled from a normal distribution, so it won't have trait value exactly ZS bar. There's some distribution around that. And here we just see the probability of establishment or reaching population size at least 100 for different values of beta. And as beta increases, actually our chance of establishing increases. And there are two things that are contributing to our chance of establishing. One of them is my migrant might be unusually fit. I'm sampling from a normal distribution, so I could just be lucky. I could just luckily sample a migrant which was maybe not of positive trait value, but at least it was close to having positive trait value. And then I don't have to do much adaptation before I'm growing as a population. Or it could be that selection's doing all the work for me, that my migrant was absolutely standard, bog standard for the source population, but selection is really, really effective at achieving something unusual and adapting to this new environment. And on the right, what we're trying to do is disentangle those two and see which is most important, or if they're roughly similar. So it's a bit of a fussy picture, but we've got, let's see. So these black dashed lines are the same as the lines over here for the values corresponding to beta equals 0.25 and beta equals 2. So that is plotted against the mean trait value in the source population. And in this case, we're both choosing the trait value of the immigrant at random from that source population, and then we're allowing the population to evolve. Now, the straight lines here correspond to a homozygous migrant. So that's one, sorry, that's the wrong way around. These correspond to fixing the trait value in the migrant. So these solid curves corresponding to choosing the trait value in the migrant to be exactly ZS bar. So I'm not allowing myself any good luck in choosing a good immigrant to found this population. I'm just letting selection do the job. Okay. And the dashed lines, I'm not allowing selection to act at all. I'm choosing the trait value of the migrant at random, but instead of having it to be a heterozygous immigrant, so having two independent copies of the alleles at each different locus, I'm choosing it to be homozygous. So it's identical at the two loci. There is no genetic variance. And so the population can't evolve. Natural selection can't act. Remember, natural selection, Fisher tells us, acts on the genetic variance. And what we see from this picture is actually both things matter. So to get to these black lines, we need and both to have good luck. So it's a combination of good luck in having a fit immigrant and good luck in actually adapting effectively. And so what's surprising is that establishment of this immigrant is feasible, even if my source population is 3 or 4 standard deviations below the threshold for growth. But the change in Z is contributed to similar degree by the fitness of the immigrant when it arrives and that subsequent adaptation. So that's the first observation. And then here's just a particular realization of the simulation to see what's happening. So this was a successful establishment from a single immigrant, which had fitness -2. It was heterozygous, so natural selection could act. And we see very, very rapid adaptation. And then every dot here represents an individual in the population. And the red line is just the mean trait value in my population as time evolves. In the middle, we see the population size. And on the right-hand side, the solid black line is the actual variance across the population as time evolves in the trait value. The line that you can't really see that's hidden, that is a wiggledy blue line, which is dashed, in fact, is sorry, is solid, is one or the other, is the actual heterozygosity. And the solid upper line is what you would predict. So I think I've written it down here. It's the predicted heterozygosity if there were no selection. So if you just looked at the population size and imagined you were sampling at random from that population, then your predicted heterozygosity would follow this upper curve here. So it sits above the actual one, which is the wiggledy blue line underneath it, but not ridiculously far above, in fact. And that's important for one of the approximations we make a little bit later on. But it does sit below it. And the reason that the true value of heterozygosity sits below it is that the pedigree is distorted by selection. It's not just going to be random mating. We are choosing parents in a way that reflects their trait value, and that will distort the pedigree that we see. Okay, but the message is the variance drops precipitously because the first generation, I'm necessarily inbreeding. I'm breeding myself. It's necessarily a selfing event because I've only got one immigrant. And it continues to drop quite dramatically. So this process of adapting, my population size is growing. This is not a linear scale. My population size is growing very nicely, thank you. But it is adapting more and more slowly as time goes on because the variance across the population is shrinking. Okay, so that was a single immigrant. So a single immigrant can establish, rather astonishingly, it can establish even when it's very unfit, but it does so at a price. So now let's suppose that we've got steady migration. And we're going to assume, for the purpose of this caricature of model, that the trait distribution across the whole population is Gaussian. Now, that, I emphasize again, is not a consequence of using the infinitesimal model, but it's a convenient approximation for us to make because it gives us a first way of understanding what might be going on. And we're going to suppose, again as a first approximation, that the population size and the trait mean and variance evolve deterministically. So now what we can see is that every time a new migrant arrives, remember what Bulmer told us. Bulmer told us that in this large source population, half of the genetic variability is within family and half is between family. So every new migrant is actually bringing half of the genetic variance in my source population. So even rather modest rates of migration into a small sink population can maintain high genetic variance. So as a very, very first approximation, we're going to suppose that the within family variance is a constant. In fact, in the paper that Nick and I wrote on this, we did something ever so slightly more sophisticated, but it was only ever so slightly more sophisticated. We did allow variance to vary, but not as much as we would have liked. So let's suppose for the purpose of today that the variance is just constant. Then remember the distribution of traits across the whole population we're going to suppose is normally distributed. That's one of our assumptions. And now I've taken the variance to be constant and the mean is going to be Z bar. And so W bar, well, I can just calculate that. That's given by this expression. And so now I'm going to be able to write down a recursion for the number of individuals in my population and the mean trait value. So the number of individuals, remember, before migration was going to be the number of parents multiplied by the average breeding value across randomly chosen pairs. Okay? That was our model. So this term is the individuals that had parents who were already living on my island. And this M is the additional migrants that I'm having in each generation. Now, what happens to the mean trait value? I've got to be a bit more careful here because the mean trait value is going to be biased. It's sort of a size bias pick from the parental trait values. So this is just normalizing by the total population size in generation T plus 1. If I look at the migrants, they've all got mean trait value ZS bar. So that's this term. And here, what I'm really seeing is the mean trait value among offspring of individuals who already lived on the island. And that is going to be weighted. So it's going to be the expectation of the trait value among parents weighted by the breeding value. So that's the expectation of Z e to the beta Z divided by W bar to make everything add up to 1. But then I'm multiplying by N of T W bar because I'm multiplying by this number, the number of individuals of that type. And so altogether, I've got MZS bar plus this quantity here, but this is calculated before selection. And how do I calculate this quantity? Well, it's easy, right? If I can calculate the expectation of e to the beta z, I can calculate the expectation of z to the beta c because e to the beta z because I just differentiate with respect to beta. Okay. All right. So now I've got a nice recursion, and it turns out I can solve it. And the right way to solve it, I want to rush through this because I can't see the time because I've turned it off on all my machines. But I'm out of time pretty much. It turns out I can solve that recursion. And what we discover is that if the mean fitness in the source population is big enough, then population size and trait can increase together. Not so surprising. It doesn't really matter what the migration rate is. But if it's too small, the population may be unable to grow really regardless of how large the migration rate is. And what I want to do is actually understand a bit more about that. So let's go, let's push on to a new model where we can understand a little bit more. So I'm going to take a continuous time approximation to my recursion because I always like continuous time approximations. And I'm going to add some noise. So the noise that I'm going to add is, well, I'll have a demographic noise because I really want the number of offspring produced by individuals to be a branching process. So this is the Feller noise that comes out of taking a branching process. And this is just saying that my branching process is not quite critical. It's slightly biased by the trait value, the average trait value in the population. And then what's this? What's this noise? Well, if I take a sample of size n from a normally distributed population with variance twice v star, the variance among the sample is going to be twice v star over n. And so this is just the sampling drift, if you like. And the nice thing about this form, so these are just the continuous analog of what we wrote down for our discrete recursion. I just prefer working with continuous models. And the nice thing about that is that we can find a potential so that the pair n and z bar take a very particular form. And for equations of this form, one can pull out one's favorite book on applied stochastic calculus and see that if there were a stationary distribution, a joint stationary distribution for trait value and population size, it would take the form e to the twice u over m. And now this isn't a stationary distribution because you can't integrate this thing. But on the other hand, you can hope that at least near a stable sink, it's going to give you a good approximation. So a long way away from that, so when we're out near infinity, we're going to expect our population size to be controlled by some sort of logistic control, say, so that we could integrate. So let's just think about what this would tell us close to a stable sink. And you see three terms. We see this term, which is increasing as migration increases. That's going to increase the population size. We see selection increasing the trait. But we see this big negative term, and this negative term is gene flow that pulls the trait mean back towards the mean in the source population, the ZS bar. And that's bad for us. We don't want that. We're trying to adapt. And in fact, if you fix the population size, you can work out what the deterministic equilibrium is for the mean trait value. And we see selection pushing us in one direction, pushing us towards being fitter, but it's opposed by gene flow, which is pulling us backwards. Integrating over Z bar, we can also find the distribution of the population size. So that's going to have a peak at a low density if the migration rate is too high. So if my migration rate is high and my ZS bar is less than about 2 standard deviations below the critical value for growth, then actually I'm going to be maintained in the sink population. I'm going to be pulled back so that I've got this big peak of population, which really isn't very fit. And what's happening is that I'm trying to adapt, but because I'm mating at random, if there are an awful lot of migrants who've just joined me, with high probability I'm choosing a migrant to mate with, and then my mean trait value is going to be the mean of my value, which I'd been painstakingly building up over generations as I adapted, and something from the source population. And that's pulling me way back again. So actually, if a population is trying to adapt a stable, well-adapted population, its optimal strategy is not to migrate as quickly as possible, because gene flow will then pull back the mean fitness. If I actually want to maintain a stable population, which would be able to maintain itself even if migration were turned off, I don't want migration to be too fast. I mean, I said nothing I said would be strictly speaking true. Ultimately, you would have a stable population because ultimately, you know, there's a positive probability of really rare events happening, but it's going to take you exponentially long before you see such a thing. So the lessons from the crude analysis is that one individual can establish, even if the S bar is 3 or 4 standard deviations below the threshold for growth. And that is a reflection of, this is Fisher. This is natural selection is a mechanism for generating an exceedingly high degree of improbability. You're producing something with a positive growth rate, even though it's starting from 3 or 4 standard deviations below that threshold. If the mean in the source population is too low, the population may be trapped in a sink. Faster migration can make matters worse, not better. It can impede adaptation and make you stuck in a sink. And the population can escape and adapt to a new optimum. And what's interesting about that is that then it will be partially reproductively isolated, and it's a model for speciation. So this is a model of speciation in spite of gene flow. And I just want to mention, this is my last sentence. I had one of these very smart Frenchmen working with me again this summer. So Sasha Rybolchenko was a master student from Polytechnique, and he did an internship with me this summer in which he extended this model to a string of colonies where you just migrate further and further away from the source population. And what we saw was that, of course, the further you moved away, the more adapted you were. And you could see a situation in which your source population is obviously maladapted. And in a bunch of islands close to the source population, you've still only got sink populations. It's not establishing an established population. But then when you get far enough away, the population is well adapted and it's not pulled back by gene flow. You actually do adapt. So this is really a more convincing model for reproductive isolation occurring. But what's scary, and this is interesting when you're interested in things like trying to assess the impact of climate change on populations that are trying to adapt, what's really scary, of course, is what we saw when we looked at a single migrant, which is the amount of variance that you lose whenever you're adapting to a new environment. And in Sasha's model, the further you move away from the source, the less variability you've got. So if your population were to change again and you were to need to adapt again, you would not have the wherewithal to do it. You wouldn't have sufficient variation left in your population. So thank you very much. I apologize, that was a bit longer than I intended, but I'll stop there.
Okay, well, thank you very much, Alison. People should feel free to unmute themselves and we can join together in applause. And even though we're over the official time, we do have time for questions if people have them. Maybe I can start with one that was coming to my mind. In the end, you were talking about different strategies for migration. So if you have a fixed population source and you want to migrate to a new destination, is there an optimal strategy for doing that? You don't want to bring everyone too soon, or if you want to, in a finite time, maximize your survival or your
in this very crude model, so you can decide what it is you're trying to optimize. In this very crude model, you could say, if I want to minimize the time that it takes me to establish an adapted population, then the answer is yes, there would be an optimal migration rate. I mean, optimize in terms of some probabilities. I want to reduce that to the minimum mean it could be, for example. But that is within, of course, this very crude model. And I should also say that as a result of a question that John Wakely asked after Nick and I gave a talk based on this last piece of this talk in Marseille a couple of years ago, he said, aha, but surely you want to think about more complicated things like dominance. And he's wasted 3 years of my life. And we do have a very nearly completed paper explaining how the infinitesimal model still applies in the presence of dominance. And these are very important biological features to incorporate. So I've presented the infinitesimal model as though it's only working for an additive trait. That's not true. It will work for more complicated traits too. And that will be a very important feature in precisely the question you're asking. But yeah, there is an optimal rate to do it. And I think what surprised me when we first thought about it was the optimal rate wasn't as fast as possible.
Mm-hmm. And it's time constant or it's varying?
Well, it depends what you're trying to optimize. If you're trying to optimize in terms of oh, I see what you mean. We only tried to work it out in the case where we had constant migration. You're right that actually one can see that you would be able to optimize further if you were thinking in these terms by reducing the rate of, let's see, how would it work? Yeah, you could get a better result by varying the rate of migration, probably by reducing the rate of migration as you once you find somebody who's sufficiently well adapted, so you stop pulling people backwards. So what you would like to do is you migrate really, really quickly until you find somebody who's used up that chance that I might have a really good migrant. And then I use my selection, my adaptation, to get me the rest of the way without getting pulled back by gene flow. But we really didn't examine that. The model is way too crude to inhale that deeply, if you like, on it. But what we would love to do, I mean, what I think this very crude analysis shows is that there is a lot of potential for actually using something based on the infinitesimal model, maybe not the full power of the infinitesimal model, to come up with some principled mathematics examining these questions instead of some combination of Barton's simulations and my attempts to make sense of his scribbled calculations. I think we could do something really nice if we could come up with a tractable way to have the infinitesimal model evolving its pedigree through time in the right way.
Thank you. Other questions? Okay. So let's just take this opportunity to thank Alison for all 3 of her lectures, which are wonderful. And I also want to thank the Irving Institute for Cancer Dynamics for their sponsorship of this and the work of the people involved, Sophie and Sion in particular. So maybe we'll have more of these at some point, but for now, this is this and Amare's lectures are it. You should be able to find them on the IICD website and more interesting activities as well. So.
And Ivan, can I add one quick thing?
Yeah.
On behalf of the IICD, I'd just like to thank you for getting this going. And also, of course, thank Amory and Alison for a great set of talks. And indeed, we will have more, and we'll get back to you all as soon as we've thought about what and when and who. So thank you very much, everyone.
Thank you for the opportunity.
Okay, have a
and
good
Simon, I'll be in touch.
Okay, sounds good. Thanks, Alison.
Okay.
Right.